So PCR amplification, I wanted to draw extra attention to this. So it's very similar to what we just spoke about with 16S, 18S, and ITS. So we have this study done in unsaturated soils here in the Chesapeake in Virginia. The idea is that we're using a novel gene to help profile viruses in soil communities. Why is this interesting? Well, viruses, unlike a lot of other organisms, do not have a 16S rRNA gene that is conserved across all members. What they do have are unique genes that are common amongst all viruses. So the majority of, there's always an exception, but the majority of viruses have capsids, right? So we can target a capsid protein gene for any type of virus and watch that in the community. So this was the focus of this study. Uses NGS technology for marker gene analysis. It helps study viral diversity. So quick background on phages. Phages, or bacteriophages, are the most abundant organism in the world. Viruses in general do infect every known living organism, but phages in general do help with cycling and nutrient profiles in lakes, in soils, in even pools, like freshwater, swimming pools. There's like some studies that do viral communities in there. So phages, they're around us everywhere. One of the first bacteriophages to be sequenced was called Phi-X. I think I mentioned it earlier. Phi-X is a very small virus. Its genome is well known, well characterized many times. There isn't a lot of repeats, so the balance of the genome is great. So we use Phi-X. It probably to this day is the most sequenced, other than maybe human, most sequenced genome because it's thrown in on every single sequencer as a balancer for library. Sampling profiles for soil. As I mentioned, we want to ensure that we're getting a good representation of the community. And so if I were to just come in here and just get one dot of this square foot, is that sufficient for this entire area? Some would say yes, some would say no. If I were to do separate extractions for all time points, or also all spatial points here, and then do different analysis there, is that sufficient? Or should a homogenization take place? So mix the sample before you extract. So really, it depends on the study. If you're dealing with root systems and trees, you'll want to do a deeper profile. If you're dealing with agricultural systems, S-pattern or the multi-plot sampling, large areas, usually 5-point sampling. So as I mentioned here, so this is a DGGE gel. So essentially, this is bacterial community fingerprint profiling. And the study here aimed to do exactly the same thing as this. This is well known in the literature. This happens in a lot of papers. We study the profile by looking at the conserved region of a specific gene. All right, and here is the exact same thing with the virus. So this is a T4 virus. Each one of these lines, each one of these lines is a theoretical T4-like phage, meaning one line, one bar here is a gene from a different virus genetically, but they are all T4-like. So this is how the researchers were able to capture the diversity of the viral profile. Quickly, so with the PCR product, you just take the PCR, you throw it straight into the constructs, do some PCR amplification or not. As Kyle mentioned,